Hello, welcome back to Regulatory Affairs 101. I am Nancy. Today, we are covering the ICHS series, the core set of non-clinical safety guidelines that support IND and CTA submissions, as well as NDA and MAA filings. If you work in drug development, these guidelines shape almost every non-clinical decisions you make. The purpose of the ICHS guidelines is simple. They define the non-clinical safety data required before a drug can enter human trials and eventually reach the market. They also harmonize expectations across global regulators, FDA, EMA, PMDA, so sponsors can design programs that meet international standards. The S series supports risk assessment throughout development. It guides study design for both small molecules and biologics, and aligns closely with ICH M3R2, which governs timing. Together, these guidelines ensure consistency and predictability in non-clinical safety packages worldwide. The S series includes 7 major guidelines: carcinogenicity, genotoxicity, toxicokinetics, general toxicity, reproductive toxicity, biotech product safety, and safety pharmacology, including QT/QTC. Let's walk through each one. S1 covers carcinogenicity studies, which are required for most small molecules intended for chronic use. The updated S1BR1 weight of evidence approach may allow sponsors to waive traditional 2-year rodent studies when justified. Otherwise, standard rodent models or validated alternatives are used to assess long-term cancer risk. S2R1 defines the core genotoxicity battery. The Ames test, an in vitro micronucleus or chromosomal aberration test, and an in vivo micronucleus or COMET assay. These studies evaluate mutagenic potential and must be completed before first in human dosing. They also support impurity qualification and early risk assessment. S3 outlines expectations for toxicokinetic sampling within repeat dose studies. PK data establish exposure toxicity relationships and support interpretation of NOAELs and safety margins. Regulators expect TK to demonstrate that observed toxicities are relevant to anticipated clinical exposures. S4 provides principles for designing toxicity studies, including dose selection, species justification, and study duration. Acute toxicity studies are often waived when robust repeat dose studies are sufficient. The focus is on scientific relevance and alignment with clinical exposure. S5R3 covers 3 segments: fertility and early embryonic development, embryo-fetal development, and pre- and postnatal development. Study timing must align with clinical plans, especially when including Women of Childbearing Potential, WOCBP, this guideline becomes increasingly important as programs move into phase 2 and phase 3. S6 applies to biologics such as monoclonal antibodies and recombinant proteins. It emphasizes using pharmacologically relevant species and allows single-species toxicology when justified. Immunogenicity is expected and must be interpreted carefully, especially when it affects exposure or toxicity. S7A defines core safety pharmacology studies evaluating cardiovascular, respiratory, and CNS effects required before first-in-human studies. S7B focuses on QT/QTc risk, including in vitro H-ERT assays and in vivo QT studies. Today, sponsors often use the integrated ICH E14 S7B approach. QT risk remains a high scrutiny area due to carcinogenic potential, making high-quality data essential. Before first-in-human studies, sponsors must complete the genotoxicity battery, core safety pharmacology, QT/QTc assessment when relevant, repeat dose toxicity studies, TK evaluations, and species justifications. These elements form the minimum non-clinical package required for early clinical entry. Before phase 2 or 3, reproductive toxicity segments and chronic toxicity studies must be completed. Carcinogenicity planning should also begin. Before NDA or MAA submission, the full reproductive toxicity package and carcinogenicity studies, unless waived, must be finalized. All non-clinical data are integrated into the overall risk assessment. Regulators expect strong species justification, especially for biologics, clinically relevant TK exposure, and dose selection that avoids excessive toxicity. Genotoxicity results must be interpreted in context, especially when impurities are involved. Common pitfalls include incomplete QT/QTc evaluation, insufficient TK sampling, poorly justified NOAELs, misaligned reproductive toxicity timing, and late carcinogenicity study initiation. For small molecules, use 2 species for repeat dose toxicity and ensure exposure multiples exceed anticipated clinical levels. Conduct genotoxicity early to support impurity qualification. For biologics, confirm target expression and consider knockout models when needed. Immunogenicity should always be interpreted carefully. For safety pharmacology, combine endpoints with toxicity studies when appropriate and ensure high-quality telemetry for QT assessments. Here's a quick summary of the S series, what each guideline covers, who it applies to, and when studies are required. Use this table as a reference when planning your non-clinical program. For more detail, refer to the official ICH guidelines listed here, along with the FDA and EMA resources. These documents provide the full regulatory expectations behind everything we cover today. Thank you for watching. If you like the content of this video, please like, subscribe, and comment below. Have a wonderful day.